Hey Mustangs, in this video we're going to be taking a look at cell membrane transport. Now what this really means is we're going to be taking a look at how molecules and materials get in and out of the cell. Now in order to really understand why this even happens, we need to remember that cells are living things. And just like when we think of yourself as the entire whole multicellular organism, we know that you need to take materials in order to maintain energy, enough energy to stay alive, and building blocks, materials to build your body, and then you also eliminate waste. Now every single one of your cells does the same thing. So just draw a little simple circle to represent a cell here. So the outside line here represents the cell membrane, and that's going to control what goes in and out. And since this is a living thing, it needs a constant supply of energy and materials going in, and it also produces waste. So every single one of your cells produces waste as well. So the things that are going to want to go in, main things are be like oxygen, nutrients, and other building blocks and materials need to go into the cell. And then things are going to need to be removed from cells too. So things like waste. So each cell produces waste that it dumps into the blood and then gets filtered out in your kidneys. Things like hormones are going to need to exit the cell, especially if those cells are part of a gland. So all of these things need to be constantly moving in and out of the cell. So the cell is not an isolated system. So things are constantly moving in and out of the cell, oxygen and nutrients and materials going in, waste and other products going out, and there has to be something that controls that, and that's what the cell membrane does. All right. Now, in our last video, we took a look at how the cell membrane is built, so the structure of the cell membrane and how that determines its function. So the function of the cell membrane is to control what goes in and out of the cell. So looking at the cell membrane here, we have just the lipids. So we see the lipid bilayer here. So we have a lipid bilayer. So that's our lipid bilayer. Okay, so we have two layers of lipids, and if you remember, the reason why the lipids arrange this way has to do with the structure where you have a hydrophilic region and then you have a hydrophobic region. So here towards the outside, let's say this is the outside of the cell. So let's say this is outside, this is inside the cell. And you have the hydrophilic regions towards the outside, so hydrophilic towards the outside, and then the hydrophobic regions face each other towards the inside of the bilayer. And the reason for that is these hydrophobic regions, they do not want to interact with hydrophilic molecules in water, while these hydrophilic regions actually do. So the hydrophilic regions are towards the outside and inside of the cell, while the hydrophobic face each other towards the middle. Now this hydrophobic region here in the middle, that is what is going to affect the permeability of the cell membrane. So you have hydrophilic, hydrophilic on the outside and then hydrophobic in the middle, and that determines the permeability of the cell membrane. So we say a cell is semi-permeable or selectively permeable. So that word selectively is key right there. So only certain things can go in and out. So we know things that are hydrophobic, such as lipids, can easily go in and out of the cell. Other large things, other things that are hydrophilic or even water itself or charged molecules and different types of waste, they can't just squeeze through the lipids to get in and out of the cell because of this region here, that hydrophobic region. That hydrophobic region does not like anything that's hydrophilic going through, does not like charged molecules going through. So because of that, these things that can't easily go through have to find another way to get in or out. So the first way that things can get across the cell membrane in cell membrane transport is simple diffusion. Simple diffusion involves simply squeezing through the lipids. So clearly, if we're talking about things squeezing through the lipids, these must be materials that are hydrophobic. So all molecules want to move from areas of high concentration to areas of low concentration. So let me go ahead and draw a picture that demonstrates what's going on with high to low concentration. So we have, I'm going to draw a super simple image of the cell membrane here. So here's my lipids, my phospholipids. Circles represent the hydrophilic region, tails represent the hydrophobic region. Here is protein, and some lipids here. Okay. Now, molecules, a high concentration means a lot. So let's say I have a high concentration here. And on this side, I'm going to put a low concentration, which means very little. So high concentration means a lot of a molecule. Low concentration means a little bit of a molecule. Because of molecular interactions, different charges and stuff like that, molecules don't like being bunched up together when there's a lot of them. They prefer to spread out. So if there is a membrane that separates two regions and there's a high concentration on one side of that membrane and a low concentration on the other, the molecules are going to want to move from high concentration to low concentration. And in the case of simple diffusion, they're going to want to squeeze through the lipids here. So in simple diffusion, the molecules move from high concentration to low concentration by squeezing through the lipids. And this requires no energy. Molecules naturally want to move this way. So high concentration to low concentration by squeezing through the lipids. So when you take a look at diffusion, molecules want to spread out. And whenever there's a membrane in between molecules, so we have two different types of molecules going across the membrane here, they always want to move from areas of high concentration to areas of low concentration. So here you can see there's a few green on this side, but lots of green here. That means these molecules would want to move to the other side. And in diffusion, what happens is molecules keep on moving to the side of low concentration until it gets about equal on both sides of the membrane. We call that equilibrium. So when they hit a point of equilibrium, so equilibrium there, when you hit a point of equilibrium, that means the molecules are going to stop moving back and forth in large amounts. So it keeps on moving until it gets about equal on both sides, and then slowly molecules will move across. So when the molecules hit equilibrium, they don't stop moving. That's a common misconception that students have. They slowly move across. So if one moves this way, one moves the other way. If two move again, it just keeps on going, trying to maintain equilibrium. All right. Then you have facilitated diffusion. Facilitated diffusion is for molecules that aren't too large, but maybe they're hydrophilic, or they are charged, and so they can't simply squeeze through the lipids because that center section there between the lipid bilayer that is hydrophobic is not going to let them through. So for facilitated diffusion, again, here's my simple cell membrane. Notice I don't draw the carbohydrates here, just the proteins and the lipids. Okay. So there's my simple membrane. In facilitated diffusion, again, this is usually for charged and hydrophobic. Oh, cross that out. Woo, you gotta be careful. Hydrophilic. So this is for charged and hydrophilic molecules. They cannot squeeze through in between the lipids. So for them, they need help. So the word facilitated, to facilitate means to help. So I'm going to draw a high concentration of molecules on this side of the membrane and a low concentration here. And molecules in facilitated diffusion still want to move. Molecules always naturally want to move from an area of high concentration to an area of low concentration. So here in this case, here's the high concentration, here's the low concentration. They can't squeeze through the lipids, so instead, they need help. And that's where proteins come in. So channel proteins and transport proteins allow a way of getting across the cell membrane. So you can think of them like a tunnel or a gate that is going to control the ability of these molecules to go across into or out of the cell. So in facilitated diffusion, molecules move from high concentration to low concentration, but they have to go through a protein instead. That's the difference between this and simple diffusion. And again, simple diffusion, those molecules are hydrophobic. Here, charged and hydrophilic molecules have to go through a protein to get in or out of the cell. Okay. So here we can see a channel protein that molecules can use to go across to slip through to the other side of the cell membrane. And then you have carrier proteins. So carrier proteins can pick up and then change shape and transport something across. Channel proteins is simply like more of a tunnel. This is kind of more like a gate. So you have to get in there, kind of use a key, and then go to the other side. Now, with these channel proteins and these carrier proteins, they're very specific. So aquaporin is a protein that allows water to go across. You can't use that for salt to go across. So these protein channels are very specific as to what they're going to let through. And usually one type of channel is going to let one certain thing through, or more than one thing, but it's still very specific. It's not going to let everything through. It's going to let very specific things in and out of the cell.